Thank you very much. Thank you very much, Professor Andrew, Professor Dr. Andrew. So thank you very much for delivering a great talk to our audience. So I would like to check whether we're going to have the any question or suggestion, comment, deficits in the live chat. Anyway, Professor Andrew, may I ask you a question? What do you expect or predict how successful the governmental actions work or cope with epidemic waves of COVID-19 pandemic in GMS, particularly in case of Thailand? Professor.
I apologize. I'm not sure I understand the question. Can you repeat that again?
What do you expect or predict how successful governmental actions work or cope with epidemic waves of COVID-19 pandemics in GMS, particularly in case of Thailand?
I can only speak for the Lower Mekong region, and I certainly cannot speak for the country of Thailand. But I think that the data is clear evidence that the government has been very, very successful in mitigating the threat of community spread. Almost all cases over the last 6 months can be attributed to people returning to Thailand, and a very, very strong system of capture of cases through a state-mandated quarantine system. And I think it speaks very, very highly to the systematic approach of not just Thailand, but Vietnam, Laos, and indeed Cambodia, in instituting what over the last 15 years they've been practicing. And again, I can only applaud really the government of Thailand for, I think this is one of the real notable successes, not just in the region, but globally.
Yes, thank you very much. Thank you very much, Professor Andy. On behalf of the organizing committees, I would like to thank for your very kind, polite talk to our audience this morning. Thank you very much, Professor Andrew. So next, I would like to proceed on delivering keynote lecture. May I invite Assistant Professor and Professor Dr. William Aldis from Faculty of Public Health, Thammasat University, to deliver a keynote lecture on whole of society intervention, myth or reality. Professor William, please.
Yes, thank you very much. I'm sharing my screen. Is it visible to everyone?
Yes. Okay.
Okay. Thank you very much. I'll start. 1 second. Okay, this presentation deals with something a bit beyond or larger than One Health itself, the larger question of whole of society interventions, which are used in One Health interventions, of course, but beyond that as well, and I will look at a broader scope. I think we should start by defining what we're talking about by framing the idea of whole of society approaches. This is a rather crowded slide, but I believe I will read it in its entirety. A strategy for addressing complex problems facing society by engaging all organizations and individuals whose participation is necessary to solve the problem, or who are impacted by the problem and will suffer consequences if it is not solved. This may include national governments. 1 second here. This may include national governments, government ministries and parastatals, multilateral organizations like UN organizations, affected individuals and communities, non-governmental organizations, including civil society groups, political parties, including opposition parties in some cases, private companies, the private sector generally, the media in all of its different platforms and forms, and sometimes, in my experience, in conflict situations, even anti-government groups, including even military and rebel groups, essential participants. What I will show in this presentation is the engagement of all these different groups is often not achieved at the necessary level of thoroughness and completeness to make a whole of society approach successful. One Health, you all know, I don't think I need to define that for this audience, but I would make 2 points. One is the collaborative, multisectoral, and transdisciplinary nature of One Health, and also the special recognition of animals, people, plants, at their shared environment, and the interconnections between them. It's certainly a fascinating area to examine. We have urban and rural humans, we have insects, we have a variety of animals, and of course we have bacterial and viral pathogens. Extraordinarily complex problem. Just in passing, this is not a presentation on One Health, but I do think fascinating things are going on. The problem of the farmed mink population in several European countries, where the minks were infected. The infection wasn't from animal to human, it was from human to animals. The minks appears to have been infected by their caretakers in these giant mink farms with millions of minks. The virus mutated within the mink and then was passed back to the humans. So the humans at one point were the vector, and at the other point, the target or the receiving organism of this infection. And it shows the complexity. And if there is not superb coordination, communication, information exchange, and joint action between the industry, the agro-industry of mink farming, and health sector, we're obviously going to have some potential risks that will emerge there. Going forward, I will leave One Health and look at the larger issue of whole of society interventions. I should probably start by saying this presentation is based on direct participation in whole of society interventions in Sierra Leone, Eastern Congo, Indonesia, and I've included 2 examples from, or derived information from 2 examples in Thailand. These are all referenced, and I've written papers on all of them one time or another. Now, so now we're back to the whole of society approach in a broader sense than One Health alone. So I should start by saying, what have I learned about the whole of society approach in these countries in Africa and Southeast Asia? We don't have time in this presentation, of course, to describe the entire process of development and implementation of One Health, of whole of society approaches in specific cases. And in any case, each country presents a different problem. However, what we can do, and what I intend to do in the remainder short time I have, is to look at markers or characteristics which tend to be present in successful whole of society interventions. And when we see failures to either achieve a successful impact or sustainability, we usually find that one of these 10 or 12 points I'll make was not characteristic of that particular intervention. So here we're talking about the difference between the aspirational whole of society approach, that is to say what we would like to do and what we might have written about and planned for, versus the operational reality. We can call this the transition or movement from the myth or the hope, the aspiration to the reality on the ground. The points I'm going to mention are by no means comprehensive. They just stand out for me after reviewing the several major interventions at national and international level I've participated in that seem to be points of success, or in their absence, failure points. So let's look at some of those. I think to start with, the particular problem in a particular context needs to be defined carefully. Now, the problem is not defining it for yourself. It's defining and understanding it collectively for individuals and organizations that come from many different institutional cultures, many different academic and scientific backgrounds, and many different sectors. If this is not done, if there's not a unifying understanding of the problem, even if viewed from those different perspectives, the implementation often goes very poorly, and people don't understand sometimes why there isn't the kind of collaboration they were hoping for. And it may be because the problem hasn't been shared to an extent of reaching convergence on what really your objectives problem is and your objectives are. And I think this is the listening process. I find very often, if I'm dealing with a rebel group, for example, in Eastern Congo, I have to listen very carefully to understand what they see as the problem, what solutions do they suggest, and also they will sometimes the least likely participant will come up with some very good answers because they may be closer to the problem on the ground. And what do they see as their role and commitment, including funds and staff? We'll come back to those last two. But everybody can talk and everybody can write an agreement, but how genuine and how sincere is the actual commitment to work on these problems in the whole society context? Here's something that's sometimes missed. I believe it's very important to build on existing programs and collaborations that are already there. It's a great advantage if you can do this. Often something in place already turns out to be crucial. I'll give the example of Thailand referred to by Dr. Corwin. In that case, we had legislation for emergency acts, emergency decrees. They were never designed for pandemics, for COVID, but they were there and they were skillfully used by the highest level of leadership of the country to take actions, which if they had not used those emergency degree provisions in existing legislation, there certainly would have been a time to introduce them on the spot. There would have been a delay in setting up some of the operational procedures. Other times, the existing platform I'm talking about here would be just on a material level. In Sierra Leone during the war, 60% of the country was in the hands of the rebels. We needed transport, we needed security for our staff, we needed trucks, we needed many things. And the World Food Program had a ship in the harbor, which became our center when the city itself, of capital city, was invaded by the rebels. Had we not had that, the helicopter that they had, the ship in the harbor, and their transport facilities, we would never have been able to react the way we did in that particular situation. Other existing networks that are so interesting to look at include the 1 million village health volunteers in Thailand, which meant that there was real-time, pre-established surveillance by 1 million people reporting to central authorities. And when a new person would come into the village health volunteers knew who they were, where they were living, and their state of health. And this surveillance system never could have been created in the real time during a COVID-19 pandemic response. It had to be present in advance. Another great example from Bangladesh I saw was the Red Cross volunteers. There were 100,000 of them. They were everywhere for communications, for surveillance, for rapid response in the flood problem, which was the one I was involved in Bangladesh for several years. These Red Cross volunteers were essential. So you don't need to invent capacity, which is already there. Always look for what's there and then invite those programs and their leadership into your whole society response. Another thing, and as a longstanding World Health Organization bureaucrat, I say this with some hesitation, but in many of these situations, you need to innovate. You need to bend the rules. You need to make exceptions. Or, and this is a secret, request other partners to do what you cannot do. In Sierra Leone, it was the non-governmental organizations that could operate, in some cases, behind government lines. In a big fight we had in Thailand over the Thai-US free trade agreement, it was the civil society aid support groups that could agitate and do very video attractive demonstrations and so forth that brought the problem of the free trade agreement, which is rather technical, to the attention of the general public, and more specifically to the Prime Minister himself, which we as a UN agency could never have engaged on that level. So you need partners on board participating, working with you, who could take actions that you cannot take. That's extremely important. And I guess this is an exception or extension of the last point. You can't do business as usual. Most organizations are not designed to work outside of their mandate and comfort zone. And I think some exceptions to that would be people in this seminar. But sometimes you need to invent new operating procedures, new funding classifications and channels. You have to move money between budget lines, and you need to be innovative. And you don't have time always to do that in a totally formal fashion. Now, this is one that came up from something that Dr. Corwin said. It reminded me of this one. Include collaborators, both individuals and organizations, with power to make real changes and drive programs. It can't just be people you like, people you worked with before. It has to be people high up. It has to be people in legislative positions. It has to be the head of state in the case of the COVID response in Thailand. Those are people that can make changes, drive programs, and you have to have that power component in your response. Finally, look for unlikely partnerships. Sometimes someone you didn't think you could work for, if you consult and go to visit that person or that organization, you find they become your closest supporters. The private sector is a good example. The provision of PPE, the donation of vehicles and telecommunications equipment, all this came from the private sector. And I would like to point out, in many cases, they lost a good deal of money to do that, but they did it because they knew it was the right thing for the country. So look for partnerships everywhere, including unexpected places. Finally, and this is, well, not quite finally, you need to commit money and people. And if those commitments are laid out and agreed on, if you don't have funds, either from existing sources or new ones, you're going to have trouble actually making things happen. You'll remain in the myth category as opposed to the reality world. And the same thing for staff. You need either full-time or full-time equivalent of staff working on whole of society interventions. The media, make them your ally, make it personal. Get to know key journalists. I found in Africa that it was useful to have biweekly lunches with key health journalists, and we knew each other well. A problem would come up, they would give me a call, I would give them a call. I could insert articles in the newspaper by just telling them I had a story they might like. It gives you access to free public messaging. And I think the relationship with media, both social media, television, radio, and print, that really solves your PR problem without you having to create a big PR system of structure of your own. And finally, I think this is not always done. You need to work your interventions on two tracks. The formal one, including MOUs, agreements between organizations, which are signed and provide stability and continuity, but also friendly, mutually respectful personal relationships where that you can make that telephone call, resolve a misunderstanding on this personal level. You need these two tracks. And finally, what I found again and again to be a secret ingredient is give credit to all your partners. I find myself on radio and TV giving speeches, and I always start out, if it's a joint effort, congratulating and giving specific examples of what everyone else has done. That's a marvelous way to increase morale and commitment of your partners. So that's all I have to say. Thank you for listening, and I hope you find it useful.
Yeah, thank you very much, Professor Wood. Thank you very much, Professor Willem. Okay.
You're welcome.
I'd like to share with our audience have any question or spec, any suggestion to you? Okay. Anyways, Professor Willem, okay, may I ask you a question? Okay. Do you have any suggestion of what is a significant role of intersector or multi-sector involvement in decision-making and the implementation of public health measures by local government of affected country in the context of COVID-19 pandemic? Professor.
Now, I'm glad you asked that because I didn't give local participation enough attention in this very brief presentation. I think that local participation is, you get a lot of answers about the nature of the problem and you get a lot of implementation power. It depends, it's so country specific depending on the way the country is organized politically and socially. But you can make, I see often a mistake made where very bright people, very capable people come in from perhaps outside the country and they work with leadership in the Ministry of Public Health or in the Ministry of Veterinary Medicine groups or whatever. But these people are all outside the problem as opposed to the people on the ground that are affected by the problem you're looking at. In the case of COVID, I think there was pretty good, partly because of the village health worker system, but not only that, there's also decentralization to the provincial level for a lot of health decision-making in Thailand. So there was a flow of information back and forth. But anybody leading a whole of society approach has spent a considerable amount of time in going to affected areas, stopping by the side of the road, talking to people, and getting that kind of feedback. So yeah, I think if you don't have local participation in the policy making and implementation, your program will probably fail.
Yes, thank you very much, Professor Dr. Willems. Very great. So, on behalf of the organizing committee, Faculty of Public Health, Thammasat University, we would like to thank Professor Dr. Willem for delivering a very kind talk today for our old audience. Thank you very much, Professor Dr. Willem. So the next session is also very special for delivering invited a lecture of this morning session to our audience. Okay, that connecting us now anywhere in Thailand or in Japan. So we're gonna have the next session, the morning session of the invited lecture. So upon this time occasion, may I invite Professor Kita Masuno from a Research Center for Zoonosis Control, Hokkaido University, to deliver an invited lecture on profiling of viral protein RNA interaction during the infection of emerging tick-borne bandavirus. Professor Dr. Kita, please. Okay.
I think you have a record of my presentation, so you can just open it.
Professor, please open video, please. Okay. Take a minute, please.
Hello, my name is Keita Matsuno, a lecturer in the Unit of Risk Analysis and Management, Research Center for Zoonosis Control. Today, I'd like to talk about our study on virus-protein RNA interaction in the cells infected with an emerging tick-borne bandavirus. Severe fever with thrombocytopenia syndrome, SFTS, is caused by a Vanda virus named SFTS virus, SFTSV, belonging to the family Phenylviridae order Viridalis. SFTS is a tick-borne disease and causes severe and fatal disease in humans. It is hemorrhagic fever-like disease, and case fatality rate in humans is up around 10% and sometimes up to 25%. This disease has been reported from Eastern Asian countries, China, Japan, South Korea, Vietnam, and recently Taiwan. A virus particle of SFTSV is an enveloped spherical virion containing 3 segmented negative-stranded RNAs. Each RNA was encapsulated by 2 virus proteins, nucleocapsid protein N and RNA polymerase L, and we call this complex as the RNP complex, ribonucleoprotein complex. 3 segmented RNAs were constituted with L, M, and S segment RNAs named based on their length. Each RNA encodes a single open reading frame of a structured protein in negative sense orientation. Only S segment RNA of SFTSB also encodes non-structured protein NSS in the opposite orientation to N open reading frame. So we call this S segment coding strategy as an ambisense orientation. After the entry of SFTSB into cells via endocytosis, RNP complex will be released into cytosol. Using the genomic RNAs in virions, initial transcription of messenger RNA and virus protein synthesis starts. Then, using this RNA polymerase and nucleocapsid protein, virus RNA replication to produce complementary RNA, cRNA, and another copies of genomic virus RNA, vRNA, will begin. Finally, using newly synthesized virus proteins and vRNAs, descendant virions will bud from cells after the assembly of the virion contents. For the SFTSV RNA replication, in addition to RNA polymerase L, RNA binding protein N is essential. SFTSN plays a critical role in virus replication by binding to cRNA, vRNA, which is necessary for RNA replication and also transcription and also assembly into cells, into virions. Our first aim of the present study is to clarify how these virus RNAs expressions were changed after the virus infection, so dynamics of virus RNAs during replication. So we decided to sequence all of the RNAs in the infected cells and compare it RNAs bound to N. For analyzing RNAs in cells, whether bound to N or not, we first inoculate SFTSV to human cells and extract their RNAs as total RNAs. In parallel, using rice cells, immunoprecipitation of SFTSV-N was performed by using anti-N monoclonal antibody to co-immunoprecipitate RNAs binding to SFTSV-N. These RNAs were called cell IP fraction. For the control of this experiment, we also performed the same immunoprecipitation and RNA extraction using supernatant of the infected cells. These RNAs were called sub-IP. Because this sub-IP fraction was extracted from the supernatant and IP'd using anti-SFTSV-N monoclonal antibody, this fraction may contain only virus RNAs, vRNAs. We expect by comparing the contents of these RNAs using next generation sequencing, we will be able to identify how virus RNA expression changes over the infection and how SFTSV-N contributes to the virus replication. We first map the sequencing width of these 3 RNA fractions to SFTSV genome sequences and human transcripts. Total RNA consisted with more than 90% of host RNAs and contained 5% of virus RNAs. After immunopurification with N, virus RNA amount was dramatically increased to 94.9%, meaning the successful RNA precipitation by co-IP experiment using anti-SFTSB-N monoclonal antibody. In the sub-IP fraction, as expected, almost all RNAs were virus RNAs, indicating no virion or SFTSB-N was released from cells with host RNA. Then we did the same experiment in the time course manner after the inoculation, and in this experiment we also analyzed the strand of sequencing reads to count each bRNA and cRNA. In the supernatant, as expected, equal amount genomic bRNAs of each segment was precipitated with SFTSB-N over the time period, suggesting the packaging of a set of 3-segment RNAs into a virion is strictly controlled. Interestingly, complementary RNAs of M segment first expressed higher than the others and becomes equal after 24-hour inoculation. There was almost 100 times difference between genomic RNA and cRNA expression in the supernatant. On the other hand, in the cells, we could observe the different expression of each RNA in both whole cell lysate and IP fraction. The difference between whole cell lysate and IP fraction of S segment is quite significant in 24 hours and 48 hours post-infection. This may suggest increasing N-free S segment bRNAs, which may be NSS messenger RNA. Similar to supernatant, expression of complementary RNA in cells decreased quickly after the infection. Taken together, immediately after the infection, replication of RNA and virus protein synthesis occurs significantly, followed by virion production period, which doesn't have so much virus RNA replication. When we go back to the initial experiment of IP and sequencing, we notice that cell IP fraction a small but enough amount of host RNAs to analyze further. Thus, we aim to reveal the involvement of those host RNAs in virus replication. We are sure some host RNAs are bound to SFTSB-N by using immunopurification sequencing analysis. So we thought that the interaction between virus protein and host RNA may have some functional role especially to the virus RNA replication. So we next map the sequencing width of cell IP and total RNA to human transcripts and calculate each expression level. Each dot in this graph represents each host transcript. Highly expressed RNAs in cells were plotted right side, and RNAs increased in cell IP were on the top of the graph area. After removing host RNAs expressed weakly, not suitable for the analysis, shown in gray dots, selected top 29 transcripts increased in IP fraction more than 50 times. These host RNAs might be specifically precipitated using anti-SFTSB-N antibody, suggesting selective mechanism of N binding to host RNAs. Then, for those 29 transcripts bound to SFTSB-N, we prepared siRNAs to knock down the expression of them. We used mini-genome system, which expresses luciferase as a reporter of virus RNA transcription, and compared with the cells transfected with siRNA controls that may not affect any host transcripts. Cells transfected with siRNAs to knock down 15 transcripts out of 29 showed lower mini-genome activity. This result suggests that these host RNAs may be important for virus RNA replication. Now, we determined in cells infected with SFTSV nucleocapsid protein and interact with specific population of host RNAs, in addition to virus RNAs. Some of these host RNAs may have functions to enhance virus RNA replication. Our hypothesis of this host RNA involvement into virus replication is that N may protect essential messenger RNA for virus replication from degradation to maintain RNA themselves or enhance virus protein expression, host protein expressions. And or N may promote cap snatching activity of virus RNA polymerase, L, which is essential for virus messenger RNA transcription. This work has done by my former student, Keita Mizuma, who is now a PhD student in Osaka University. And I'd like to say thank you for all the members who are involved in this study. Thank you so much for listening. And I'd like to take any questions.
Yes, thank you very much.
Oh, my name is Keita Matsuno, a lecturer in the Unit of Risk and...
Thank you very much, Professor Keita. Yeah. So I would like to check whether we're gonna have any question or suggestion to your talk. Okay. Anyway, may I ask you, I would like to ask very simple question than you talk. So SFTSV has a 2 cycle. Is this correct? One is a synthetic cycle, another or urban cycle. That is correct.
Hey, could you repeat that question again? Sorry.
Okay, Professor Masuno. My question is very simple than you talk. So very simple. I would like to know SFTSV has a 2 cycle. One is synthetic cycle, another urban cycle. That is correct.
Do you mean as like dengue virus or something?
Virus. SFTSV virus has 2 cycle, synthetic cycle, and another urban cycle. That is correct.
I don't think it's not the case for this virus.
Okay. And normally these virus can develop an epizootic cycle, right?
So do you mean the virus can replicate in stick cells?
Okay. So how successful this virus SFTSV epizootic cycle occur in many amplifying hosts, for example, sheep, goats, cows, or even domestic cat. Is it possible or not?
Ah, I see. So because this kind of tick-borne virus has a very simple structure as a virus, so they only have a 2 protein for the virus replication.
Okay.
And it works both in the tick cells, animal cells, and including human cells. So the host specificity may not be restricted by the function of the protein.
Okay. Yes, thank you very much. Thank you very much. On behalf of the organizing committees, Faculty of Public Health Thammasat University, we would like to thank for your delivering talk. Thank you very much, Professor Masuno. Thank you. So next, I would like to proceed on delivering invited lecture. May I invite Professor Dr. Katsuro Hakiwara, School of Veterinary Medicine, Veterinary Virology, Rakunokaku University to deliver an invited talk lecture on the relationship between hepatitis E virus and the host. Professor, please.
I'd like to express thanks to organizer of this meeting for the opportunity to make a presentation. Today, I'd like to talk to you about the zoonosis as a One Health topic. As shown in the title, hepatitis E is a zoonotic disease that infects both animals and humans. From the characteristic of this virus, the One Health concept is important for the prevention of this disease. Hepatitis E HIV can be summarized the following points according to the WHO reports. Hepatitis E is a liver disease caused by infection with hepatitis E virus. Every year, there are estimated 20 million of HIV infections worldwide, leading to an estimated 3.3 million symptomatic cases of hepatitis E. Every year, mortality cases of HIV infection are reported. The virus is transmitted via fecal-oral route. Hepatitis E is found worldwide. A vaccine is not yet available elsewhere. Now let me explain the characteristics of this virus. The virus belongs to Hepatidae, has no envelope, and has single-stranded RNA as its genome. This viral genome encodes at least 3 open reading frames, where ORF2 encodes the capsid protein. ORF1 and 3 encodes several molecules as shown in the slide. So far, several major viral genotypes have been isolated. Next, when the virus classification is organized, the susceptible hosts of each genotype are classified. Of particular note today is also herpesvirus A, genotype 3 and 4. It is organized as a zoonotic disease and is distributed worldwide. On the other hand, genotype 1 and 2 are transmitted human to human. The slide is a diagram showing the genotypes in the phylogenetic tree. In this way, genotype 3 and 4 virus are transmitted between humans, livestock, and wild animals. Some of the virus from animals are still unknown to the transmission to humans. HIV are distributed all over the world, as you can see in this slide. The countries shown in red are regions with the highest incidence. Now let's talk about the virus transmission. HIV is transmitted mainly through the fecal-oral route. The risk factors are related to poor sanitation, ingestion of undercooked meat or meat products derived from the infected animals, transfusion of infected blood products, organ transplants, vertical transmission from the pregnant woman to her baby. Epidemiological studies of HIV in Thailand have been reported, and the results showing the regional characteristic. It was 30% positive from the northern to central parts of Thailand, but low in the southern part. Area with many Muslims do not eat pork there. That means pork consumption might be one of the causes of the human infections. We analyze the VGM maximum credibility based on the HIV genome sequence obtained from an epidemiological study of pigs in Vietnam. As a result, it was shown that the pigs are shown as a reservoir of HIV, but humans also affect HIV infection as a reservoir of this disease. It is a graph showing the amount of the virus shedding from the serum and the feces in the experimentally infected piglets. In genotype 3 and 4, a large amount of virus is shedding from feces immediately after infection, with almost the same tendency in the both genotypes. The shedding virus is not only the source of infection of other pigs, but also a source of the environmental pollutions. Well, this is a liver of pigs. Which liver is infected with HEV? Perhaps it is indistinguishable at a first glance. Infection cannot be distinguished by observation organs. In the HE staining of tissue, small cell infiltration is observed in the infected livers, left-hand side. Therefore, infected pig cannot be detected by livestock inspections. This is a result of the pig shedding in feces from birth to the end of the fattening in pig farming. We are comparing the average number of the virus copy in the feces of piglets born from 2 litters. As shown in the figure, when the two groups are combined after weaning, the infection spread at once, and the virus continued to be shedding in the feces for 70 days or more. The peak at the time reached 10 to the 6 per gram of the feces. In this way, virus spread and environmental pollution are established. When the host immunity is sufficiently established, the virus shedding disappears. But the liver at the finished stage is still detected in 20 to 30%. That is, viruses reinfection occur from fattening to the final stage. Actually, it has been reported that the virus was detected in liver sold in grocery stores in the city. In this study, many genotype 3 are confirmed in those livers. Let's talk about the HEV transmission. Piglets are infected with the virus from contaminated environments and infected soils. After that, it spread to the other piglets by shedding the virus in the feces. Infected pigs do not show clinical symptoms. As it is fattening and used an ingredient of the meat as a meat. In many cases, people get infected from undercooked meats. Unfortunately, hepatitis occurs. Now, I'll talk about the iatrogenic HEV infections. This is a number of post-transfusion HEV cases confirmed in Japan. Even today, patients infected with HEV by blood transfusion are confirmed. This figure showing the infection rate of the blood donors. On the left is a number of the positivity of gender. The feature is that there are overwhelmingly many men. On the right is the detect of HEV genotypes. This is only characterized by many genotype 3. In Japan, mainly genotype 3 were detected in donated blood. Now, next is the graph showing the HEV infected blood donors around the world. Even in the country with well-developed infrastructures, there are HIV positive donors. Many of them donated blood without clinical symptoms. Blood donors with high ALTs, alanine aminotransferase level have been found in a higher proportion of hepatitis carriers. Among these hepatitis, the only zoonotic disease is HIV. Unfortunately, some patients progress to hepatitis due to liver transplantation. As shown in the graph, seroconversion has been confirmed. In addition, it has been reported that post-operative HIV infection persists due to immunosuppressive drug treatment. Now, I'll summarize the circulation of HIV. HIV can be transmitted from sow to piglets or from environmental HIV to piglets. Once infected with piglets, the infection spread among the piglets. HIV is retained in the farming. If infected drinking water or infected meat is undercooked, the virus will be transmitted to human by eating. Some people develop to hepatitis, but some are asymptomatic with viremia. If that viremic person donated their blood, HIV-infected blood will be donated, and infection will be established by transfusion to other patients. HIV nucleic acid screening of donated blood may be necessary to prevent infection. This viral infection is associated with all elements of animal environment, human medical care. There is another important characteristic of the virus. It is heat inactivation. The virus is easily inactivated by heating in PBS. However, inactivation is difficult even in 60 centigrades when heated with albumin, which is a blood component. The HIV inactivation requires heat treatment at a temperature higher than 60 centigrades. At the end of this presentation, I'd like to make the following suggestions. One health concept is important for zoonotic disease prevention, such as the HIV. Please consider measure on the assumption that you already have a lot of infected livestock and wild animals. Infected meat is marketed, has no regions. Infected pigs shedding the virus in feces for a long time needed to be careful environmental contamination and transmission to others. The HIV infection cycle similarity to other zoonotic disease. Therefore, it is a good idea to consider the countermeasures using this infectious disease as a model. I'd like to thank you all the collaborators who participated in this study. That is all my presentation. Thank you very much.
Yes. Thank you very much, Professor Hakiwara. Thank you very much. Okay. We would like to share with, this section has any question or suggestion, comment. I would like to check first. Okay. You have the 1 minute left. So can I ask you a very simple question? Professor Hakiwara, could you please provide more information about HIV prevention and vaccine?
Hi, thank you very much for giving the talks. And regarding the vaccine, up to now, there is no vaccination program right now, and then no vaccine are developed, have not been developed, and the only Chinese studying a vaccine prevention, but we don't have no chance to have such kind of vaccine. Maybe we should think about more progress regarding the vaccine product.
Yes, thank you very much, Professor Hakiwara. So on behalf of the organizing committees, Faculty Public Health, Thammasat University, we would like to thank you very much for clearly delivering your great talk. Thank you very much, Professor Hakiwara. And next, I would like to proceed our invited lecture. May I invite Professor Dr. Pirom, Faculty of Public Health, Thammasat University, to deliver invited lecture on cell-mediated immune response to avian influenza H5N1 virus. Professor Pirom, please.
Thank you very much for giving me an opportunity for sharing my research project. The topic today is about the cell-mediated immune response to avian influenza H5N1 virus. So as we have already known that the human influenza A virus, not only human influenza A virus, subtype H1N1 and H3N2, but also avian influenza virus such as H5N1, H7, and H9 can also infect to human. So this slide shows the timeline of avian influenza virus infection in human. You can see that the H5N1 virus has emerged at the first time in 1996 at the China, and then the H5N1 virus caused the human infection. The first case report is around 1997 in Hong Kong, and then the virus re-emerged again in 2003 in Hong Kong, and then the virus continued to infect humans until to present. And the important one is about the S5 and 6 has emerged in 2014, and then this virus also infects humans. But normally the H5N1 virus has caused the high fertility rates in humans. So now we identify that the H5 and H7 subtypes are highly pathogenic avian influenza virus. Okay, this slide shows the cumulative number of confirmed human cases for avian H5N1 virus report to the World Health Organization. So you can see that since 2003 to 2019, we have the total case around more than 800 cases, with the more than 400 cases were died. So the fertility rate is about over than 50% in Thailand. Thailand officially report avian flu outbreak on 23rd January 2004, and there were 25 human avian influenza virus H5N1 cases with 17 deaths. The last case was seen in July 2006. The last event of protein die-off was reported in the lower north in 2008. However, at the beginning of the 2020, we found the outbreak of the H5N1 in poultry in China. So it means that the comeback of the H5N1 virus to human may be possible. So we have already known that when we got the infection, the influenza virus infection, or we receive the vaccination, two arms of the adaptive immunity have developed and less again to the virus. The first one is the antibody-mediated immunity, that we know that the antibody against the HA and NA are classified as the act as the neutralizing antibody and protective antibodies. And the second one is the cell-mediated immunity that involves the T-cell response. The T-cell response is recognize the immunodominant epitope that rely on the viral protein. If the T cells recognize the HA or NA, that it means like a subtype specific T cell response, but sometimes T cells directly recognize to the conserved epitope that rely on the NP, M, or the polymerase protein. So the T cells can be called that the heterosubtypic T cells mediated immunity. So the scope of the study, we recruit the subject, the two types of subject. The first one is the H5N1 survivor. For subjects, we collected blood samples, the sequential blood sample, every 6 months of the 4 H5N1 survivors. And the second group is the non-H5N1 subjects, around 33 subjects, including 10 subjects of the H3N2 patient. So the acute and convalescent blood samples were collected. And 23 healthy individuals, we collect the blood sample for healthy individual also. So this study divided into two parts. Part one, we detect the humoral immune response. It means that we detect the neutralizing antibody against the H5N1, H1N1, and H3N2 subtype. And the second part is we study the cell-mediated immune response by using the ex vivo interferon gamma ELISpot assay to identify the H5N1 specific and cross-reactive T cell epitope. And also we perform the flow cytometry to determine the immunophenotypes of the T cells and the function of peptide-specific T cells. The result of part one is show you about the neutralizing antibody against influenza virus in H5N1 survivor. This table shows you about the demographic data of the survivor, the survivor number 1, 2, 3, and 4. And we collect the sequential blood and test and detect the neutralizing antibody. We found that all of our 4 H5N1 survivors has markedly high antibody titers against H5N1 virus. In addition, we perform that these survivors contain a neutralizing antibody against the H3N2 or H1N1 or not. But we found that they also contain a neutralizing antibody against the seasonal H1N1 and H3N2 virus also. And we detect the neutralizing antibody against influenza virus in non-H5N1 subject. We found the H3N2 patient, they contain the neutralizing antibody against the H3N2 and the H1N1 subtype, but they did not contain the neutralizing antibody against the H5N1. Again, the healthy subject also contain the neutralizing antibody against the H3N2 and H1N1, but they did not contain the neutralizing antibody against the H5N1. So it means that this group, non-H5N1 group, did not contain the neutralizing antibody against the H5N1. And the second part, we detect the T-cell mediated immunity. This shows the step of the study by using the ELISpot and flow cytometry. We isolate the peripheral blood mononuclear cells from the H5N1 survivor and the non-H5N1 subject. So the PBMC samples were subject for the first round ELISpot assay by using the H5N1, NP, M, and HA overlapping peptide to determine the magnitude of the total T-cell response, and we detect the matrix analysis for preliminary identification of reactive peptide. So we get the candidate peptide. After we get the candidate peptide, we confirm by using the second round ELISpot assay for confirmation of specificity of the reactive peptide using the individual peptide stimulation. And then after we have already the specific peptide, we establish the cytotoxic T-cell line like effector cells or T-cell lines by using that epitope. Again, we perform the functional assay. Before we perform the functional assay, we have to establish the autologous Epstein-Barr virus transform B lymphoblastoid cell line, or we call TBCLs, as the target cells for functional assay. So if we have already T-cell line, effector T-cells, and the antigen presenting cell like TBCL cells, so we perform the functional assay by ELISpot and flow cytometry. ELISpot assay, we detect the interferon gamma secreting T cells, and flow cytometry. Flow cytometry, we detect the immunophenotype, and we perform the intracellular cytokine staining to detect the interferon gamma, TNF alpha, as well as the upregulation of CD107A granulation marker. This figure shows about the concept for designing overlapping peptide. Overlapping peptide spanning NP, M, and HA protein derived from the H5N1 isolate from Thailand. The first Thailand isolate that come from our laboratory. So this peptide contain 20 amino acids. You can see that the peptide number 1 contains 20 amino acids. Peptide number 2 contains 20 amino acids, but peptide number 2 contains overlapping 10 moles to peptide number 1. So after that, we perform the peptide pool matrix. So this shows the example for the NP peptide. The total of the NP peptide is around 49 peptides in total, and then we perform the peptide pool matrix, so we can get the 14 pools of NP peptide, like a pool A1 to A7 and pool B1 to B7. And then if we get the peptide pool, so we perform the ex vivo interferon gamma ELISpot assay. Interferon gamma ELISpot assay, this show in this slide. So the cell stimulation, we add the PBMC into the reactive well, and then if the PBMC specific to peptide can recognize the peptide, so the T cell can secrete the interferon gamma. Interferon gamma can be detected by this system. So at the right hand side, show the ELISpot interpretation. So you can see the purple spot in the reactive well. one spot, it means the one T cell which secrete interferon gamma. So we can count the number of T cell response. This shows the examples of the result obtained from the first round spot assay using the overlapping peptide pool. So you can see the purple spot in the reactive wells. It means that one spot shows the one T cell response we can count, and then we include the negative control that we did not add the PBMC. We did not add the peptide, and we perform the positive control by adding the phytohemagglutinin for positive control. And this slide show the timeouts, right? Not yet. Okay. This slide show the total magnitude of the T cell response against H5N1, NP show in A, M show in B, and HA show in C. So you can see that the survivor number 1 and survivor number 2 show the total magnitude, show the higher total magnitude of T cell response than the survivor number 3 and survivor number 4. And when we comparing the response against the NP, M, or HA, we can see that the T cells higher response against NP than M and HA. So it means that NP is an immunodominant protein for recognizing by T cells. And this shows the second round ELISpot assay. We confirm that which peptide can be identified. So we can identify that the survivor number 1, we identify 5 peptides, 2 peptides from NP, 2 peptides from M, and 1 peptide for HA in the survivor number 1. And also survivor number 2, we can identify the specific T cell epitope. And again, after we identify epitope in survivor, we identify epitope, the cross-reactive T cell epitope in the non-H5N1 subject. The results show that we obtain the cross-reactive T cell epitope identified in non-H5N1 subject, and we show that the the STN2 patients show the higher ex vivo interferon gamma response higher than the healthy subject. And also we established the autologous IBV cell line and the T cell line and test for functional assay by using the ELISpot assay and flow cytometry. This slide shows the clump of polyclonal T cells that we call the effector T cells. And then we put together between the T cells, effector T cell, and EBV B cell line that act as the antigen presenting cells and as with the peptide we call the pulse peptide. And also we perform the flow cytometry by intracellular cytokine staining. We detect the cytokine production like interferon gamma, TNF alpha, and CD107A, and detect the immunophenotype, which the CD8 or CD4 T cells. So you can, the results show that the CTL effector T cell when meet to the peptide pulse antigen presenting cell, so the T cells secrete the interferon gamma and TNF alpha. And also the other system, we use the recombinant vaccinal virus containing the NP. So it shows that the NP is like stimulate the T cell response also. And this shows the CTL, the polyfunctional T cell. It shows that the T cell that we developed by using the in vitro stimulation show the secretion of the interferon gamma, TNF alpha, and CD107A expression together. So it means that the T cell that we obtain act as the polyfunctional T cells. And this slide shows the the epitope that show the cross-reactive T cell epitope and the specific T cell epitope. So for this question, we can obtain the subtype specific and cross-reactive T cell epitope, and we show that NP is more immunodominant, and show that the longevity of H5N1 peptide specific T cells lasts for at least for 4 years after disease onset. And also, finally, we propose that the boosting of T cell-based immunity by using a novel vaccine against conserved epitope would provide a high level of immunity with broader degree of protection across influenza subtype, in particular a pandemic strain. So we would like to thank Ajahn Sasithorn Theptakarnpon, Dean, Faculty of Public Health, Professor Pilaipan, Professor Orasa, Professor Uraiwan, and the management team of the Faculty of Public Health and my colleagues. Thank you very much. Sorry for over time for my presentation. Sorry.
Thank you very much, Professor Dr. Birong. Okay, due to the time constraints, so on behalf of the organizing committee, Faculty of Public Health, Thammasat University, we would like to thank Professor Dr. Birong for the highly provided talk to our audience this morning. Thank you very much. So I would like to proceed on the other invited lecture for this morning section. May I invite Professor Dr. Manapon Wolfsmanchai, Faculty of Public Health, Thammasat University, to deliver an invited lecture on microplastic contamination in aquatic animals in the Chao River estuary. Professor Dr. Manapon, please.
Thank you very much. Good morning. My name is Manapon Wolfsmanchai. I come with the topic of microplastic contamination in aquatic animals in the Chao River estuary in Thailand. Nowadays, we face on the microplastic problem because the million ton of microplastic entering to the surface water every year. Thammasat University by the Faculty of Public Health start this project because we concerned about the microplastic in our country. First of all, I would like to inform you all that this is the preliminary study of the microplastic in the Chao River Basin. Okay, now let's see what is the microplastic. Microplastic is the plastic particle less than 5 millimeters in diameter. It can be separated into two types. The first one is primary microplastic, means plastic directly released to the environment in the form of small particulates. For example of this type of microplastic is microbead, such as in the toothpaste, or plastic in the nail polish. For the second type is the secondary microplastic. It means the microplastic that originated from the degradation of larger plastic items into the smaller plastic, such as the bottles, the plastic bag, when it degraded or it cracked by wind or wave or sunlight. Where microplastic come from? There are several sources of microplastic. For example, municipal wastewater is one of the significant sources of microplastic because microplastic is from the personal care As I mentioned before, the synthetic textile is the significant source of microplastic in the municipal wastewater because it accounting for 35% of the microplastic released to the water. Industrial wastewater is one of the source of microplastic pollution. And the next one is runoff. The abrasion of tire while driving is the significant source of microplastic from the runoff. And the plastic that degraded in the landfill is also the source of microplastic. The use of fertilizer that made from the sewage sludge is the source of microplastic from the agriculture, and the shipping and recreation activity are one of the microplastic source as well from the marine coating. When the microplastic from all sorts released to the water, the aquatic animal ingest them and they pass through the food chain and eventually to the human health. The adverse effect of microplastic can be from two ways. The first way is from the microplastic itself. This is because the additive that added to the plastic, for example, the palette, the BPA, these substances are added to the purpose to the plastic for the purpose. For example, the BPA is added to make hard plastic, but this substance is dangerous for human health. The next way is from the substance that absorb to the microplastic, such as the PCB, pesticide, POPs, substance, or heavy metal. These have the dangerous for human health. As I mentioned before about the source of microplastic and the danger of microplastic, the Chao Phraya River Basin is the study area because all activity that provide the source of microplastic are in this area. Looking at this picture, the Chao Phraya River is the main river of Thailand and is the main river of Thailand. It's cover around 20,000 square kilometers. In cover 9 provinces of Thailand. It has the agricultural area in the upstream and downstream. There are agricultural area, municipal area, and several industrial estates in here. And the downstream, they have municipal area and industrial area. And this it actually has the aquaculture and recreation area for the people.
บางอันมันไม่มี reference แบบดีๆก็เอาแบบนี้ reference ดีๆก็ปะชื่อปะแล้ว
So
นี่มันเป็นแค่รูปภาพแบบไม่มีอะไร
So Chao Phraya River Basin has high risk of microplastic contamination. Since this is the preliminary study, we select some of aquatic animals to analyze the microplastic with the objective to indicate the contamination of microplastic in fish and shrimp from the Chao Phraya River estuary and to identify the type of contaminated microplastic. The area that I circle is the sampling point. I collect 4 type of the 4 species of the animal: croaker, boatman croaker fish, shrimp, and sea catfish. And then all of the samples were taken to the laboratory. First step to analyze the microplastic is the sample preparation by rinse it with distilled water and extract and separate the meat and entrail. After that, we extract by the hydrogen peroxide and heat them around 55 to 65 degrees Celsius for 24 hours. After that, we separate the microplastic with the sodium chloride and filtrate it with through the filter paper. After that, we dry it, the paper filter in the oven around 4 hours and analyze for the physical property by the stereo microscope, and then confirm for the polymer type with the Fourier transform infrared spectroscopy, and from now on I would like to say the FTAR. The results show that the crocodile has the highest number of microplastic contamination, followed by the Bozeman croaker, shrimp, and sea fish. The result also show that the shape of the microplastic is the pellet or granule that you can see in the slide, and the fiber I provide for two pictures for the fiber, and foam here, and fragment is here. For all shape of the microplastic that we found in the study, fiber is the main shape of microplastic. It's accounting for 46%, followed by fragment, foam, and granule. This study also revealed that the microplastics are accumulated in the entrail higher than in the meat. You can see that the microplastic accumulate in entrail about 74% and in the meat about 26%. This slide shows the ratio of the microplastic accumulate in entrail and meat by the species. You will see that the shrimp had the highest ratio of microplastic contamination in meat. Is about 42% of microplastic that accumulate in the meat. But the sea catfish had the lowest ratio of microplastic that accumulated in the meat because it's less than this. Okay, the result from the FTAR show that the type of the polymer, we found that polystyrene, polypropylamide, polyethylene, and nylon are the polymer type in the microplastic. Then we track back for the source of the polymer type. We found that the polystyrene is made for the container. This may come from the electrical cable and wire, and polyethylene come from the plastic bag, and nylon is from the fishing net and rope. Although the result from the preliminary study revealed that the aquatic animal in Chao River Basin contaminated with microplastic, but more data are needed to support this study. Therefore, next plan for this project is to analyze the microplastic in water and sediment, and analysis of microplastic in more species and more sample size. Thank you very much.
Yes. Thank you very much, Professor Dr. Manapon. Due to time constraints, so we, on behalf of the faculty, organizing committee, faculty of the Thammasat University, would like to thank for delivering very kind talk this morning to our audience. So thank you very much, Professor Dr. Manapon. So I would like to inform all the audience and our distinguished speaker and invite speaker for our morning section of the program. So we want to close the morning section. We will have the lunch break. Okay, 1 hour's lunch break. So the local time in Thailand now is 11 a.m. and the local time in Hokkaido is 13 p.m. So we will have the 1 hour lunch break and we'll come back here again. Start the afternoon section. We will have the 3 invited speakers before we have a next afternoon break. So please join us, okay, for our afternoon section. Thank you very much. See you. Okay. Oh, I got some message, maybe very interesting, because we also have the open live chats, allow any participant that receive their question or suggestion comments. Our distinguished speaker and vice speaker will be asked to check whether there are any questions specific for your session. Please respond, because we think we're going to have a question or a suggestion deficit now. Thank you very much. See you soon, very soon. Thank you.